Hi, my name is Kathy Olson, and I'm here to tell you about novel ways to monitor cell growth and cytotoxicity using our StainFree technology from molecular devices. Today's presentation covers the StainFree technology and applications, including cell counting, monitoring cell growth over time, cytotoxicity assays, expression analysis, and viral plaque assays. We'll also discuss the Sectormax IT multimode system. StainFree Technology is Molecular Devices' patent-pending algorithm for counting cells and estimating confluence without the use of fluorescent dyes. It's available on the Sectormax i3 plate reader with the Minimax imaging cytometer. So it helps users to eliminate time-consuming fluorescent dyes and to be able to monitor cell growth without actually disrupting the cells, and also to set up imaging assays all within the Softmax Pro software. The StainFree technology eliminates cell staining for cell counting and confluence measurements using a proprietary transmitted light analysis technology. Basically, users can take bright field images and have their images analyzed by the software. To have the software count cells, you simply draw the outlines of the cells in the images and then select background areas. So the software automatically calculates segmentation parameters. If the results are initially not as expected, further teaching can be done to refine the results. An option in the software would be to use predefined settings that are included for many cell types. In the software, we indicate where cells are by drawing with the cell tool, which is shown in yellow, and we indicate where the background is by drawing with the blue tool, shown as a blue squiggle on the image on the left. So the blue background tool can be used to indicate areas between the cells that we don't want to have counted as cells. It can also be used to draw on various artifacts, small bits of dust and things like that, or scratches on the bottom of the well that we want the software to exclude and not count as cells. After drawing on several cells to indicate them, and also drawing on several areas of background, we can click Apply and then see the results. If the results aren't immediately acceptable after the first round of training, we can go back and do further drawing on additional cells and background areas to refine the cell identification. Once the training is complete and we're satisfied with the results, we can view them, and the cells will show up as found objects, which are shown as purple masks in the software. Now we're ready to read or reanalyze the entire plate using the settings that we've created. The StainFree cell counting works with a wide variety of cell types. So here I'm showing 3 cell types, the HeLa, U2OS, and RBL. The HeLa cells are more refractile and smaller, where the U2OS are more flat and spread out. And then the RBL cells grow in suspension, so they're small and round. But the software is able to handle all of these different cell types. And in fact, we have predefined settings that will work for the cell types shown here, and also a variety of other cell lines that have similar morphology. So there are some cell lines where it may be very difficult to distinguish the cells by eye, and where that's the case, it's also difficult to train any software to recognize individual cells. So that's the case with the cell line I'm showing here, which is HepG2. These cells tend to grow in clumps, and within those clumps it's very difficult to identify individual cells looking at a bright field image. So we do offer an alternative way to analyze these cells in the software, and that's called field analysis. So instead of identifying individual cells, this field analysis can be used to identify areas that are covered by cells, in other words, confluent areas, as an alternative to counting discrete objects or cells. So the image on the bottom shows the confluent areas that are identified by the software for the HepG2 image. One very useful application that we can do with the StainFree technology is a cell growth assay. So in this example, HeLa cells were seeded at 4 different initial densities. Those are at 2,000 cells per well, 1,000, 500, and 250 cells per well in a 96 well plate. The cells were then imaged using the TL, or transmitted light channel, at intervals for 7 days. And then the percent area that was covered by cells was measured using the stain-free analysis, using the field analysis option. So the graph is showing for the 4 different initial densities of cells, the growth patterns that were observed over those 7 days. So you can see on the x-axis is the time in hours, and that's the time post-seeding. And on the y-axis is the percent area covered by cells. So I think this is a very useful application that can allow users to monitor their cell growth, for example, if they need to let cells reach a certain confluence level prior to performing another type of assay on them, say a compound treatment or something like that. We can also use the stain-free analysis to do a cytotoxicity time course. So in this example, the HeLa cells were seeded at 3 different densities, 4,000, 2,000, and 1,000 cells per well in a 384 well plate. And those 3 densities are represented in the plots that are 3 different colors in the graphs to the right. So I'm showing graphs that were from data analyzed at 16 hours, 40 hours, and then 72 hours. And within each of those graphs, the red plot represents cells that were seeded at 4,000 per well, the green is for 2,000 per well, and the blue plots are for the HeLa cells that were seeded at 1,000 cells per well. So at time 0, these cells were treated with N-isomycin, which is a compound that induces apoptosis by inhibiting protein synthesis. And over time, the cells were monitored using the stain-free analysis, and we were using the field analysis feature for this particular experiment. So again, at 16 hours, we can see that the IC50 curves are starting to develop. So as we look at on the left-hand side is low compound concentration, and moving from left to right, we have higher compound concentration. So you can see curves starting to develop even after 16 hours of monitoring. And then at 40 hours, our curves are getting more distinct and steeper. And then by 72 hours, we have very pronounced IC50 curves. And the IC50 values calculated from these curves are shown in the table at the bottom left. So you can see that there's not a huge change in IC50 for any of the cell densities over time. They pretty much stay similar for this particular compound. So it's an interesting way and a useful way to be able to monitor cytotoxicity without having to lyse the cells or stain the cells or do anything else that may disrupt them, and we can monitor them as long as we want to. So in addition to time course type assays, we can do cytotoxicity comparisons between different compounds. So I'm showing here an IC50 curve for N-isomycin in red, and also trichostatin A in green. And trichostatin A is another compound that induces apoptosis through a different mechanism. So these cells were treated with the 2 compounds for 72 hours, and the initial seeding density of the HeLa cells was 1,000 cells per well. And after 72 hours, I measured the percent area covered by the cells treated with different concentrations of the compounds, and then plotted the IC50 curves in Softmax Pro software. So again, the IC50s are shown in the table below, and you can see the difference between the 2 compounds very clearly. We can use the stain-free analysis to do assay quality control. And in this example, HeLa cells were treated with staurosporine to induce apoptosis, and that's shown in the top set of images. So they were seeded at 3 different densities, 2,500 cells per well, 5,000 cells per well, and 10,000 cells per well. And the top row of images is showing the cells that were treated with 37 micromolar staurosporine, which is a very high concentration that induces the apoptosis after 4 hours. And then on the bottom row of images, we show the untreated cells. So they have a more flattened and viable looking morphology. So we use the stain-free analysis to determine the percent area that was covered by the cells. And then those same cells were further assayed to detect caspase activity. And caspase is an enzyme that gets activated as part of the apoptotic pathway. So we used the luminescent caspase activity on the same cells and took that signal, the luminescent assay signal, and normalized it to the area covered by cells. And so the bottom graph is showing you those normalized results for each of the densities of cells per well. And you can see that they're pretty consistent from one density to another. So I feel this could be used as a kind of assay control measure to control for things like unevenness of seeding of the cells. One kind of assay that's often done using fluorescent dyes is expression analysis imaging. So in this example, we're going to show how you can use the StainFree technology to identify the cells, and then within each of those cells, analyze the expression of a fluorescent marker. So in this case, CHO cells were labeled with a red mitochondrial dye, and we tested 4 different concentrations of the mitochondrial dye to simulate different expression levels or changes in expression that might be induced by a treatment of the cells. So the cells were imaged using the transmitted light and the red fluorescent channel. So transmitted light was to pick up the individual cells so that we could count them, and the red fluorescent channel is to pick up the signal coming from the mitochondrial dye. We identified the individual cells by stain-free analysis. So again, this eliminates the need for a nuclear dye since we can identify cells purely from the TL images. And then we calculated the average intensity of the red fluorescence per cell. So in this way, without having to use any kind of nuclear marker, we can get a calculation of expression or intensity of fluorescence per cell using the TL to identify our cells. Here is a summary of the expression analysis from the previous slide. And this again is looking at the 4 different concentrations of the red mitochondrial dye that were applied to the cells. And we're showing that we can detect different levels of fluorescent mitochondrial labeling and distinguish that on a per cell basis. So on the y-axis of this graph is average intensity of red mitochondrial dye signal per cell. And then on the x-axis is the concentrations applied for the dye. So again, no nuclear dye was required to identify individual cells here. Here's an example of a type of assay that typically requires a lot of time and hands-on effort to complete. So it's a plaque assay. A viral plaque assay is used to determine virus concentration in terms of infectious dose. And the traditional method would involve infecting a layer of cells with the virus and then looking for plaques or cleared areas where the cells have been infected under a microscope and then counting those plaques or infected cell areas manually. So that's very time-consuming. It often involves having to use a stain. So we can do that using the stain-free analysis method. In this example, cells were grown in a 48-well plate under an agarose overlay containing substrate. And then the cells were infected with virus expressing the beta-galactosidase. And when the cells are infected, they form blue-colored plaques, which were visible in the TL channel of the Minimax cytometer. To quantitate the plaque assay, the stain-free analysis method was used to identify the plaques. So in the software, using the yellow drawing tool, we're able to identify what is a plaque. And then with the blue drawing tool, we're able to identify to the software what is background. And that's shown in the image on the right. Once we've trained the software, it can correctly identify plaques in the images. So the image on the left is showing the transmitted light image of plaques. And then on the right, we see the purple mask indicating the plaques that the software has identified based on our training. So it takes a tedious and time-consuming process with a lot of hands-on and converts it into a mostly automated process that takes a fraction of the time. To summarize, I've shown you a variety of ways that we can use the stain-free technology, including cell counting, monitoring growth curves, assessing cytotoxicity, normalizing plate reader data, and identifying viral plaques, all without the use of dyes. I'd like to wrap up this presentation by telling you a little bit more about the Sectormax i3 multimode detection platform. And that's the instrument that was used to generate all the data that I just showed you. It has 3 detection modes built in, and those are absorbance, fluorescence, and luminescence. And more modes can be added to the system as the scientist's needs evolve. So in the lower left-hand corner of the slide, I'm showing the instrument with its cartridge drawer open. In that cartridge drawer, you can actually install additional modes in the form of cartridges, which are shown at the bottom of the slide. So just a few of these I'm showing are HTRF, alpha screen, and fluorescence polarization. So those can be purchased and installed at any time after acquisition of the base unit. We also have a Scan Later cartridge, which allows users to scan and detect Western blots on the same platform. And then the orange part of the instrument in the lower left is actually the Minimax imaging cytometer, and that allows the user to add at any time imaging capability to the base system. The Sectormax Minimax imaging cytometer can be added onto the i3 system at any time, and that adds cellular imaging capabilities to the plate reader. So it allows you to combine cellular imaging with microplate assays into a single system, allows you to count cells and measure confluence without the use of dyes using the StainFree technology, which I just told you all about. And it has two-color fluorescence, so you can expand the cell viability and toxicity applications that are available. And all of this is done within the SoftMax Pro software, which is used to acquire the data and the images and to do complete analysis. Thanks very much for your attention, and if you need more information, please visit us at www.moldev.com.